lesson in biology will be on protein synthesis. We will cover the areas of translation and transcription. Both of these processes involve important things such as DNA and RNA.
I'm bored. I hate these movies. The actors are always so bad.
Whoa.
Guys, come on. We need to learn about protein synthesis. We can always watch another movie.
Anything!
Okay. Protein synthesis. What's really going on?
What is tRNA? What is rRNA? Where does protein synthesis occur? Cytoplasm? The nucleus? What about those genes anyway? They're trying to just make a fashion statement? What's DNA? What is up with that stuff? What is protein synthesis?
The world never knew until now.
No, stage left. You've got it all wrong. Protein synthesis is a process in which proteins are made based on the codes instructed in our DNA.
Anybody know what this is? Anybody? Can anybody tell me what this is? Does anybody think this is important? If you don't think that previous image was important, then you should. Is deoxyribonucleic acid, better known today as DNA, genetic blueprint for all living things. Contains all the information, control all body processes, and controls all the cells in your body. It also contains the code for the making of proteins, which is transcribed onto mRNA.
DNA is made up of 4 nitrogenous bases: adenine, thymine, cytosine, and guanine.
In DNA, adenine always bonds with thymine, and guanine always bonds with cytosine.
This is a model of a section of DNA. The corresponding nitrogenous bases, adenine and thymine and cytosine and guanine, are connected by hydrogen bonds, which are very weak. The nitrogenous base pairs are connected to the sugar molecules, which are held together by phosphates.
To describe the transcription process, you must know about RNA, or ribonucleic acid. It's similar to deoxyribonucleic acid, DNA, except that in RNA, it has ribose as the sugar instead of deoxyribose. And there are 3 types of ribonucleic acid. There's mRNA, tRNA, and rRNA, where mRNA is the messenger RNA, tRNA is the transfer RNA, and rRNA is the ribose RNA.
During transcription, the DNA unzips at the hydrogen bonds in the nucleus with the help of the enzyme RNA polymer.
In this section of unzipped DNA, the mRNA, represented by this strand here, enters the nucleus, and it uses the unzipped DNA as a template for its matching strand. And as you can see, the thymine here bonds with adenine, and guanine bonds with the cytosine. But over here, as you can see, this is adenine, and in transcription, thymine is replaced by another nitrogenous base called uracil, and uracil bonds with adenine.
Once the mRNA is finished with transcription in the nucleus, the mRNA travels to the ribosome, where the process of translation begins.
Hey! Have you seen tRNA?
I think he's over there.
Thanks!
Bye, mRNA!
Bye!
Hey, mRNA!
tRNA!
What's up?
Nothing, dude.
Yeah!
Translation time!
I'm just so upset.
Have you heard anything about your missing proteins?
Got the letter in the mail today.
A letter? Can I see it? It looks like some kind of code. I think I know what it means. We should take this to my friend tRNA. He's a really good translator.
That's a good idea.
Hey tRNA, we need your help.
What do you need help with?
We found this mysterious letter. It's in some sort of code and we don't know what it means. Can you translate it for us?
It's not a very common code. I think I can do it. let me look through my code book to see which one it is. Ah, here it is. Just what I needed. The wheel of amino acids.
Dun dun dun!
Well, this shouldn't take me long, so be back quick.
2 hours later.
Eureka! Got it!
What's it say?
Let's see. It says, Dear Catherine, thronine, arginine, stop. Serine, 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 phenylalanine, glycine, theronine, serine, arginine, stop. Glycine, proline, serine, glutamine, tyrosine, proline, leucine, serine, arginine, stop. It says that.
But what's it mean?
Yeah.
Oh, it just means you've been invited to a party.
Oh, see.
I'm just kidding. There's no party.
Aww.
What I actually did was I just decoded the codons from your mRNA sequence for your missing proteins. What I actually did was decode the codons in the mRNA sequence. And a codon consists of 3 consecutive nucleotides, in this case, adenine, cytosine, and guanine, and each of these signifies a specific amino acid, in this case, thronine, which can be added to the polypeptide chain, which is a sequence of amino acids. Chain of amino acids, or polypeptide, ending with a stop codon, signifies one protein.
So how did you do it?
Easy when you have a chart like this. You start with the first letter in the codon. It's A. The second letter may be C, and if the third letter is G, the amino acid you have is 309. So the codon, made up of 3 nitrogenous bases, is in the ribosome, and that also codes for an anticodon, which is attached to the tRNA. The anticodon matches the nitrogenous bases with each other, and it goes out and grabs free amino acids, which attaches to the chain of polypeptides.
Let's see. As the tRNA, I have to go get serine. Next, I have to get the glycine. Stop. Legit, stop.
It's time for a song.
Turn it up.
I think it's the end.
sum up, the DNA contains all the genetic code, which is comprised of sequences of nucleotides. And the DNA controls all cell activities and even make proteins. And this is called protein synthesis.
The first step to protein synthesis is transcription. Transcription is when the DNA code is transcribed or copied into the messenger RNA code at the nucleus. Thymine is not a base in RNA. It is replaced by uracil. The mRNA carries the code from the nucleus to the ribosome, which is located in the cytoplasm. Transcription ios is complete.
The second step in protein synthesis is translation. In the cytoplasm, at the ribosomes, the DNA code is translated to a sequence of amino acids. There are 3 tRNA bases, which equal 1 anticodon. 1 anticodon matches 1 amino acid. Transfer RNA, or tRNA, anticodon carries amino acids from the cytoplasm to the ribosomes and matches its complementary code on the mRNA. The amino acids are linked together to form specific proteins. Scientists in Berkeley, California have uncovered key new information towards understanding the crucial first step in protein synthesis, the process by which genetic code harbored within DNA and copied in the RNA is translated into the production of proteins. This new information also helps to explain how viruses such as hepatitis C are able to hijack protein synthesis machinery in humans for their own purposes.
Our information was obtained from the research archives and the science archives at Berkeley Lab, California, on the information of the protein synthesis and hepatitis C infections, and also from the Prentice Hall biology textbook on Virginia edition, copyright 2004, and also from the teachings of Mr. Halte, our science teacher.
This video was brought to you in part by
Katie! Julia!
Also,
Special thanks to
Jacob.
guys, good luck on the SOL test.
Don't forget to get a good night's sleep
And brush your teeth.
and
don't
for goodness sake, eat your freaking Wheaties!